I hope you are doing good. In this section, we will be talking some questions about cell and organelles. Usually, as far as MBBS first year is concerned, the long type of question which you usually get is you can get a question on mitochondria, because mitochondria has its own DNA, but that usually is taught under DNA. So we usually try to ask only questions related to lysosomes again, or the structure of the mitochondria. Usually you get it as a class test type of question. In this, a long question we do not expect. Small question here and there, lysosomes, peroxisome, mitochondria can be asked. But as far as MCQ is concerned, mostly because these questions cannot be asked in the long term type of question, or even you can't ask to write a short note on lysosomes, peroxisomes, mitochondria, because as far as in the final examination is concerned, you have lot of important topics to be covered in the examination. So usually these type of topics are covered well in MCQs. So let's get started. We are now starting with the MCQs on cell and its organelle. The question number 1. Which cell organelle is responsible for primary packaging of chemicals that will be dispersed throughout the cell? Now, the packaging of the chemicals, or the packaging of the biomolecules, or the packaging of the proteins, and further its targeting, whether a protein will be targeted to the nucleus or to the mitochondria or to the peroxisome, or it is going to go into the cytosol, or it is going to go and land up in the cell membrane or outside the cell membrane. The correct answer is yes, you are all correct. It is Golgi apparatus. It is Golgi apparatus. So Golgi apparatus is known as the site for cell sorting. It is the site for cell sorting or packaging, right? Or we also call it protein targeting. Protein targeting, because protein will be targeted to the various cell organelles with the help of the Golgi apparatus. Then the second question, the inside of the cell contains a jelly-like substance known as, now see, there is a term here, ectoplasm. Ectoplasm is the region of the cytosol on the outer side towards the cell membrane. So when you say ectoplasm, it is part of the cytoplasm only, and it is the cytosol where the majority of the pathways are occurring. For example, cytoplasm is the site where the enzyme, where the glycolysis ka first half takes place, that is conversion of glucose to pyruvate take place. It is the site where your most of the pathways are present. Fatty acid synthesis is going to take place here. The protein synthesis also takes place here. By and large, all the anabolic processes, this is not a universal thing, but by and large, most of the anabolic processes takes place in the cytosol. The catabolic processes and the anabolic processes are divided on the basis of the compartment, so that the two processes do not occur at the same time. Whenever there is synthesis occurring, only synthesis should take place. The intermediates should not intermingle and should not be confused with those which are going to form whether it is going to go into breakdown or synthesis. So for the clarity, what nature has done is compartmentalization. Now, question number 3. Answer this question. Name an organism with a single chromosome and cell division via budding or fission. So this is Usually it is discussed only for prokaryotes. The prokaryotic cells, they are the organisms which are having single chromosome. They have cell division, they are doing budding, and they can also fuse. Then this process is only occurring in prokaryotes. So the correct answer to this question is prokaryotes. Inside our body, even when they infect our body, still they continue to do budding. So they replicate, their mode of replication is by simple budding. Then question number 4, which organelle is involved in maintaining the cell shape, providing the structural support? It is done by the cytoskeleton. Cytoskeleton forms the framework. So it helps in the maintaining the structure of the cell's shape, providing the structural support. Here, question number 4, the answer is A. Some of us are giving wrong answers. So make sure that you know that whenever you have to maintain the cell shape, you have to have a cytoskeleton. Like skeleton maintains the body shape, similarly cytoskeleton will maintain the cell shape. It is A. Which of the following is cell's outermost boundary? On the outside, exterior most is always present plasma membrane. So plasma membrane is the part of the outermost part of the cell. You have a cell, on the outermost part is present plasma membrane. It is the plasma membrane which interacts with the exterior of the cells. So it protects the intracellular contents from going to the outside. So it is plasma membrane. Yes, you are all correct. What is the mechanism by which water passes through a semi-permeable membrane from a location of a higher concentration to a region of the lower concentration? What is this process known as? Answer this question. Answer question number 6. It is A. Yes, it is A. Osmosis. See, when you use diffusion, diffusion occurs for the solute. When you are using the term osmosis, it is for the water. So when water is passing through a semi-permeable membrane, the process is known as osmosis. It will always move like the solute. Solute moves from a region of its higher concentration to a region of its lower concentration. This is diffusion. This is diffusion, right? Now question number 7. What is the name of the process by which cell consumes the material across the plasma membrane? Now this here is, what do you mean by cell eating? It is endocytosis. It is trying to, better term for cell eating will be phagocytosis, but here we do not have phagocytosis. Here we have a term endocytosis. It is taking in inside. So endocytosis is a process by which cell will take the material across the plasma membrane. So endocytosis takes inside the cell. Exocytosis outside the cell, right? Now there are various ways by which cell communicates on the outside. Now this we will study it in detail under cell membrane, and the topic for the cell membrane in detail will be covered in other video. The questions on the cell membrane will be covered in another video. But what cell does is, one of the major ways by which cell takes in the material is endocytosis, and by the major way by which it exits is exocytosis. Then question number 8. Glucose 6-phosphatase is the biomarker for the following subcellular entity. Now when you say answer question number 8, this is little trickier. Now this, try and understand. There, for every tissue you have a marker. For example, if I say there is somebody is having MI, there will be necrosis. Necrosis will release the content of the heart outside. That means there will be release of CKMB, troponin I, troponin T. These are the markers of the tissue. Likewise, we also have cell organelle ka marker. That means that particular organelle is having that particular enzyme. It is not present anywhere else. Now, here I did not get one correct answer. Glucose 6-phosphatase is the enzyme which is present only and only in liver endoplasmic reticulum. It is a marker of endoplasmic reticulum. It is not a marker of cytoplasm. Most of the students are saying cytosol. No. Now, for cytoplasm, the marker is lactate dehydrogenase. For cytosol, the marker is LDH. That means you will find LDH only in the cytosol. For plasma membrane, the marker is 5' nucleotidase or sodium potassium ATPase pump. There can be more than one marker. It should be easy to detect. For Golgi apparatus, we have galactosyl transferase, manosyl transferase. These enzymes are found in the Golgi apparatus. They are responsible for cell sorting. They help in sorting of the proteins. The protein will undergo modification, that is done by these enzymes. Then for peroxisomes, the marker is peroxidases, catalase. Then for nucleus, the marker will be DNA. For the nucleolus, the marker is rRNA, ribosomal RNA. It is the most abundant RNA and it is synthesized in the nucleolus. The marker for nucleolus is rRNA. So these are major cell organelles. Mitochondria. For mitochondria, mitochondria is powerhouse of the cell. There is an enzyme pyruvate dehydrogenase or ATP synthase. These are present only and only in mitochondria. They are found only in mitochondria. For lysosome, the markers are various hydrolases. You have a long list of hydrolases. They are present only in lysosome. For example, sphingomyelinase. This enzyme is found only in lysosome. Then you have many enzymes. There is hexosaminidase, aryl sulfatase A, aryl sulfatase B, beta glucuronidase, beta glucocerebrosidase. There are various markers of lysosomes. More on this under the topic sphingolipidosis. Sphingolipidosis. Alright, answer question number 9. Name the process by which vesicles membrane fuse with the plasma membrane and the contents of vesicles are excreted in the surrounding media. What is this known as? We have said this earlier. It is known as exocytosis. Exocytosis is the process by which the content, yes, you are all correct. It is the process by which the contents of the vesicles are produced outside. They are released outside. Then question number 10. Answer question number 10. Which of the following cell organelles has a prokaryotic cell-like appearance? Prokaryotic cell-like appearance. In the humans, there is a cell organelle mitochondria which has prokaryotes-like appearance. Like in prokaryotes, there is a closed circular DNA similarly is present in the mitochondria. Mitochondria also is known to have a closed circular DNA. So yes, the correct answer is C. The mitochondrial DNA is discussed in detail under DNA after you have covered your molecular biology. Then it will make much more sense. But mitochondria, probably it is said that during evolution, mitochondria was a prokaryotic cell which got inside the eukaryotic cell because it is like a functional cell. Mitochondria behaves as if it is a functional cell. Then question number 11. Answer question number 11. Which of the following types of cells, in which of the following types of cells the gap junctions are absent? I am waiting for your response. Question number 11. The correct answer is yes, you are right. It is sperm cells. The sperm cells are motile cells. They are individual standalone cells. They don't need gap junctions. Gap junctions is required for those cells which are staying there, which have a fixed position so that they can communicate with the adjacent cells. So sperm cells do not have, any motile cells do not have gap junctions. Then question number 12. Which of the following cell organelles is responsible for storing food and other nutrients that a cell needs to survive? What is your answer for question number 12? It is vacuoles. Vacuoles are storing, yes, you are all correct. It is storing food and the nutrients which the cell will require. They are formed from the endoplasmic reticulum. So they will have all the vacuoles are formed inside the cytosol. They are present and they carry all the nutrients. Question number 13. Which of the following cell organelles is found in animals but is absent in plant cells? Plant cells do not have centrosome. During mitosis, the mitotic cell division requires centromere formation, and this is where that occurs only in animal cells. We saw it earlier that prokaryotic cells, they divide by budding. The mitotic spindle formation and division, that occurs in animal cells. Suicidal bag is referred to lysosomes. Any macromolecule is there, it will come to the lysosome. That macromolecule will keep on accumulating in the lysosome. It will never be converted to any micromolecule if the lysosomal hydrolases are absent. So the purpose of lysosomal hydrolases is to convert that macromolecule into micromolecule. Now, most common disorders that affects the lysosomes are if these enzymes are deficient. Then what will happen is there will be accumulation of these macromolecules, and such disorders are known as lysosomal storage diseases. You will study many disorders under sphingolipidosis. Lysosomal storage disorders include mucopolysaccharidosis, There is a glycogen storage disease, which is also Pompey's disease is also categorized under lysosomal storage disease. Which of them is involved in phospholipid synthesis? Now, there are two types of endoplasmic reticulum. One is smooth and another is rough endoplasmic reticulum. The smooth endoplasmic reticulum is the site of lipid synthesis, cholesterol synthesis that occurs in the smooth endoplasmic reticulum. Rough endoplasmic reticulum is the site for protein synthesis. Option D is not correct, or would have been correct if option A would not have been mentioned, because particularly it is stating smooth endoplasmic reticulum or microsomes. Even if that would have been mentioned in option A, that is correct. So whether you call it smooth endoplasmic reticulum or microsomes, it is one and the same thing. Where do cisternae belongs to? Cisternae are part of the Golgi apparatus. Now, the synthesis of the proteins, they occur at the level of proteins are synthesized on the ribosomes, which are attached to the endoplasmic reticulum, that is rough endoplasmic reticulum. From here, the protein does not go to the cytosol. It goes inside the Golgi apparatus. It will release cisternae. It has cisternae, from where it will be releasing the proteins to the various cell organelles. Whether a protein is going to destined to the nucleus, mitochondria, peroxisome, cytosol, cell membrane. So Golgi apparatus decides cell sorting or protein targeting. That we discussed in one of the previous questions. Question number 17. rRNA is produced in. It is produced by the, there is a nucleolus. It is not having its own cell membrane, but it is a region inside the nucleoplasm which is rich in chromatin. Chromatin-like fibers are present there. It is the site of the rRNA synthesis. There are two types of rRNA, prokaryotic 70S, eukaryotic 80S, further divided into smaller and the larger fragments. I am sure you know this. These are further made of 16S, 18S, you have 23S, 5S, 28S, 5S and 5.8S. It is the 28S rRNA in eukaryotes and 23S rRNA in prokaryotes which form the peptide bonds. They are said to have peptidyl transferase activity. In other words, they are rRNA that acts as ribosomal RNA that acts as enzyme. Hence, they are examples of ribozyme, RNA acting as enzyme. The 18S rRNA recognizes the cap on the mRNA, and the same function is done by 16S rRNA in case of prokaryotes. There is no cap in case of prokaryotes, but there is a sequence known as Shine-Dalgarno sequence that is recognized by the 16S rRNA. So two rRNAs, different types in prokaryotes and eukaryotes, depending upon the sedimentation coefficient. S is sedimentation coefficient. More on this in detail under RNA topic. Then question number 18. Which of the following promote curvature of the cell membrane? The cell membrane is made of phospholipids. The phospholipids are on the outside are different from the phospholipids on the inside. On the outside, we usually have phosphatidylserine, and on the inside, we usually have phosphatidylethanolamine on the outside and phosphatidylserine on the inside. Now, there are two leaflets. There is an outer leaflet and there is an inner leaflet. The outer leaflet is rich in phosphatidylethanolamine. See, all the phospholipids are part of the biological membranes. They are present on the outer side, phosphatidylethanolamine, on the inner side, phosphatidylserine. So, the curvature is produced by the phosphatidylethanolamine. But if phosphatidylserine is present on the outer side, that is on the outer part of the membrane, then we call it marker of apoptosis. Then it is known as marker of apoptosis. Question number 19. Which of the following cell type has sarcoplasmic reticulum? The answer is muscle cells. The endoplasmic reticulum found in the muscle cells are unique. They are modified and they are rich in calcium. Anywhere motility is occurring, calcium is required. If the vesicle is moving, calcium is required. If the muscle is contracting, there is movement of the myosin, troponin, actin, fibrils, you require calcium. So motility is done by calcium. So muscles are having calcium in it and it is stored in the endoplasmic reticulum, which is modified modified function of the endoplasmic reticulum is found in the muscle cells, known as sarcoplasmic reticulum. Which of the following are the phagocytic cells? The phagocytic cells are, answer this question, they are neutrophils and macrophages. Yes, it is neutrophils and macrophages. These are phagocytic cells. These cells can phagocytose foreign substances like bacteria and they can kill them because the phagocytic cells, they have an enzyme known as NADPH oxidase. This enzyme converts oxygen to superoxide free radical, and if this enzyme is deficient, you will find the disorder. So that will be your homework. What is deficiency of NADPH oxidase called? The superoxide free radicals can kill the bacteria in the macrophages. So phagocytic cells, they have this enzyme so that they can kill bacteria. Alright, question number 21. Lysosomes are produced by which of the following cell organelles? They are formed from the Golgi complex. Lysosomes themselves are derived from Golgi complex. There are certain enzymes which will reach lysosomes only if Golgi complex will be directing them to the lysosomes. Such enzymes are known as lysosomal hydrolases. If these enzymes never reach the lysosomes, what will happen? The macromolecules will keep on accumulating in the lysosomes The macromolecules will keep on accumulating in the lysosomes. They are not going to be converted to their micromolecules because enzymes never reach none of the enzymes reached not one, their multiple enzymes did not reach the lysosomes. Such disorder is known as I cell disease. I stands for inclusion cell disease. Inclusion cell disease. These are accumulation of macromolecules. They are included only in the lysosomes. They keep on aggregating in the lysosomes. Which cell organelle is responsible for the synthesis of phospholipids? What is the marker of the endoplasmic reticulum? I think you are able to tell me now that endoplasmic reticulum ka marker is glucose 6 phosphatase. Right. So this you have to know. Who is the father of cell biology? It is George Emil Pellade. He was the father of cell biology. He did many research work telling us about where the protein synthesis is occurring. He studied in detail the ribosome. Whatever we now know about the ribosomes, it is his contribution mainly. Question number 24. Protein synthesis takes place in which of the following cell organelle? The site for protein synthesis is, yes, you are all correct, it is ribosomes. Particularly those which are attached to the endoplasmic reticulum, because a free protein is never released directly into the cytosol. It is modified by the Golgi apparatus and then targeted to the various tissues. Perfect. Now this is basically on one of the extracellular matrix questions. Just to give you some insight, we are mainly talking about cells. Just one here and there question on extracellular matrix. Of course, that topic will be dealt in detail later. A 29-year-old female presents to her psychiatrist with concerns that she may have OCD. She explains that she becomes extremely obsessed with making sure that her fruits, vegetables are sanitized by rinsing with water exactly 60 seconds, then boiling for 60 minutes. She refuses to eat any fruits or vegetable that does not undergo this process, which she has began doing about 3 months ago. Now, this kind of OCD many people have developed, particularly after COVID-19 infection. Which of the following vitamin deficiencies is most likely she is going to develop? It is vitamin C deficiency. Vitamin C is required by an enzyme, prolyl hydroxylase. This enzyme is required for the formation of collagen, the post-translational modification of collagen. Collagen is part of extracellular matrix. If there is no vitamin C, there will be no strong collagen. Collagen will not be very stable, and that will result in scurvy. And that is why we see bleeding gums in vitamin C deficiency. Please note, cooking particularly destroys vitamin C. Alright, and hence my answer is vitamin C. Thank you all. Keep studying.